Thank you very much for the introduction, and thank you for the invitation to speak today. It's a real privilege to be here, and I'm really looking forward to meeting and speaking with many of you over lunch. But before that, let me tell you about some of my research. I have to admit, I'm not the world's expert on remyelination, but I'm going to tell you how I came to actually work in that area indirectly through my collaboration with Kayleen. So in order to get there, I'm going to try the clicker, which works. So I'm a neuroscientist, and I'm really obsessed with the brain. I think it's the most interesting organ in the whole body. It controls not only how our body functions, but it plays a key role in who we are. And I'm sure you're more aware of that than I am, because when something happens to your brain, it changes your ability to be who you are, to interact with other people. And while you may retain an essence of who you are, that interaction and the in and out between the inside and the outside world can be compromised and can become very challenging. So in order to try and understand that and find a solution for people who do have brain disorders, my goal is to understand in some tiny, tiny way how the brain works. So I'd just like to introduce you to some concepts around how I think about the brain, and my research is mainly in brain plasticity. And the reason why brain plasticity is so important is because it's everything we do, everything we learn, the actions that we do, the experiences that we have, those things all change our brain physically. They change the way the brain actually is organized. And in order to understand that, you need to think that the brain is not a filing cabinet. It's not a place where you put little drawers and a memory gets filed away and packed up in a corner, and when you need it, you go back and you find it and you pull it out again. In fact, if you take away from all of the tissues and the cells in the brain, you're left with these amazing networks and connections, much like those in the room today, that extend way out into the whole world. And it's those connections that carry the information. So our memories are not stored in little packages. They're stored in networks and in pathways throughout the brain that allow us to then follow those pathways and retrieve memories and store experiences that can then be retrieved as well. So the slide that I'm showing you at the bottom two pictures are essentially those pathways that are very important to us today, because they're the ones that are often myelinated and that are so key in carrying information throughout our brain. So when we talk about plasticity, what is brain plasticity? I've mentioned before that it's about that ability to change those pathways and to store information. But let me talk about it perhaps through a simple analogy. So there are some parts of our brain that are really busy and they get used a lot. And for those of us speaking today, it's our little stress centers where we have our anxiety and worries and fears and fears that we're not going to be able to get words out. But those parts of the brain can be really, really busy, just like a very busy highway in a large city. There are other parts of the brain that are very simple because they have one thing that they need to do, which is, for example, to get information from the brain down to the other parts of the body, or from the eye into the visual parts of the brain. And those are just straightforward pathways that deliver information to one particular target. The brain is always under construction, though, just like a city. You can have the busy parts that become busier, so you need to build new buildings, new roads that allow that information to be passed around and transmitted more effectively. If you have a very simple path, for example, in a developing brain, that brain region needs to be elaborated, so you have new roads that dive off it, that become branched and form more connections with other places. But equally, in all of that plasticity event that's going on throughout your life, sometimes you can have injuries where areas are disconnected from each other. And in this example of a public transport map where unfortunately a train had to undergo track repairs or whatever, there are ways that you can get around that. So the origin and destination might be detached, might be separated, but if you put in bus routes, you can still get around. It might not be as convenient, we all hate buses, but it is still possible to bypass the area of injury, and the brain's very, very good at doing that. And this is essentially what the benefits of brain plasticity are, is that they allow you to overcome the problems that stop the brain from working the way that they should. So in an ideal world, we'd like to be able to harness that plasticity. And we know that it happens all the time, and we know that it can have massive effects on the way the brain works. So you've probably all heard that blind people can compensate for their blindness by amplifying their other senses, so they become more sensitive to sound and to touch. And you can actually visualize this in the brain. And the images here, for example, you have a sighted person in an MRI scan at the top, And this person is in an MRI machine and is feeling an object in their hand. And when they feel something, that touch sensation is represented in the bits of the brain that are in red, the somatosensory cortex. If you put a blind person in an MRI machine and give them the same item, they feel it not only in their somatosensory cortex, but the visual cortex, the bit that normally would process vision, has actually been repurposed, because there's no point in having a visual cortex if you're not getting any visual input. So the brain has been able to reorganize and bypass those areas that don't work to be able to make sure that what input is coming in is processed to the best of their ability. And this happens obviously in younger people and in children to a great extent, but also in the adult. And there is a sort of myth around saying that as you get older, your brain plasticity decreases. And I'd like to just temper that a little bit with some recent research that shows that when you get a young person to imagine doing a motor task in the MRI machine, or even to actually do something, so putting objects together when they're in an MRI machine, these are the parts of the brain that they use. And you can see it involves both hemispheres, it's sort of the motor area. But then when you put an older person in the MRI machine and get them to do the same task, they actually use many more areas of their brain. And we don't really know why, but it does give me hope that even as we age, our brain can undergo a lot of plasticity to perhaps compensate the bits that aren't so efficient and make everything work well. We may have to work a bit harder, but our brains can still support the same tasks that we did when we were young. So how can we harness this brain plasticity to use it in contexts where perhaps the brain may not be working so well, to bypass lesions, for example, in MS, and to compensate for structures that aren't working? So the city here is represented the beautiful city of the future, the city where all of our brain is in harmony, where we've got a beautiful balance and great connections between all the different regions. And we know this idea of use it or lose it. So the more you use your brain, the better things are. And physical activity, I'm not arguing with anybody, that's probably one of the best interventions that you can have to maintain brain health and brain plasticity. But that's not available to everybody. I personally hate exercise, and I'm sure a lot of you in the room, perhaps that's not something that you are able to do to an extent that will actually generate the chemicals and biochemical processes in your brain that allow you to maintain brain health and brain plasticity. So my research is focused on alternatives to those things, and I'm very excited to talk to you about brain stimulation, which is a way of indirectly activating the brain by using external stimuli. So many of you may know that thousands of years ago, the ancient Romans actually used brain stimulation, and they did this by using electric fish that they would put on top of the head of people who had very severe headaches. And it wasn't just the head. People would put electric fish on parts of the body that had gout or inflammatory diseases. And to a certain extent, I believe that those processes were more or less effective. And those practices have fortunately come a long way since those days. We no longer use electric fish to deliver stimulation to the brain. Instead, we've been very fortunate that understanding of physics has allowed us to avoid that sort of direct electrical stimulation, because we know that that's very painful. If you get an electric shock, it can be dangerous, it's difficult to control. Instead of using that direct electrical stimulation, we can use magnetic stimulation. Because if you think back to physics that you might have learned in school, magnetic induction is really powerful because it can bypass all of the skull and the scalp. And once that magnetic field hits the conductive tissue inside the brain, it can start to induce electric currents in the brain that essentially activate brain circuits just like they do when we exercise, when we have very exciting thoughts, when we have great experiences out in the world. So these brain stimulation techniques are really, really powerful to target particular brain areas. Most of the research in this area has been carried out in the area of depression, because anxiety, depression, those sorts of behaviors have long been known to be susceptible to these electrical treatments. Many of you may be thinking of electroconvulsive therapy, which still remains one of the most effective treatments for depression. However, this particular treatment, which is called repetitive transcranial magnetic stimulation, is something that can be delivered in a non-invasive and completely, I won't say completely, but it is not painful. You can feel a muscle twitching, you can feel your muscles perhaps getting a little bit stiff, but it is delivered when you're awake, sitting in a chair, and you can stand up at the end and drive home. So this is a really exciting alternative to those electroshock treatments, which can be a lot more severe and have a lot of side effects. So this treatment has been approved for depression around the world in various countries, and just last year, 2 years ago now, 2021, has been listed on the Medicare benefit schedule. So it's now an alternative treatment for people who don't respond to antidepressants and benefit from this sort of direct brain stimulation. However, these sorts of treatments can be very variable, and the point of this slide is basically to show you that depending on a range of factors that can include your age, your genetic background, like Nick and Anna talked about just now, all of those things can influence how well you might respond to RTMS, this transcranial magnetic stimulation. So while diversity in people is a wonderful thing, it makes it really difficult to do the sort of research that I do, which is trying to understand how the brain works, because we all have very different brains. I'm going to talk a little bit about my research in some animal models, because these animal models can be developed in such a way that they're very uniform. So it helps us to really get down to the mechanisms of what's going on in the brain, and we can then identify the different factors that we think will then help us understand what's going on in humans. So I've told you a little bit about my background in research and how I work in brain plasticity, and you're probably all wondering, well, when's she going to talk about remyelination? You know, how do I get there? And I think this background that I'm telling you about is really because it's the beginnings of the Taurus 2 trial that you'll hear about later on, but these are some of the real basic concepts that came out of the work that I've done with Kayleen, and that led almost serendipitously to the sort of work that is now affecting people's lives. So I'm going to talk, first of all, about how science is really fun and unexpected. I do research, and every day I arrive in the lab, I have no idea what's going to happen. And sometimes these questions come up, and you have to go down these very strange pathways. So I'll share a few stories about that. I'm going to talk about some of the animal models that we've studied in depression, but then also how that relates to MS, and show how sometimes when you're working in one field, that information can then inform another, and you end up with incredible new insights that can go beyond a simple question that you started off with. We'll talk very briefly about some of the outcomes that we've had and where we want to go in the future. And then I'll just end with some thanks for all of the people in my lab, obviously, who really did all of the work that I'm privileged to talk about today. So, science is fun and unexpected, and I'm sure many of you know that. Oh, my clicker's gone. Thank you. So these are the sorts of equipment that we use when we do TMS in the clinic to stimulate the brains of people with depression. And my goal was to try and understand what was going on, because this technique was developed almost sort of by a physicist talking to a doctor, and they said, let's give it a go, and they started stimulating people's brains. And it hasn't really been through the rigorous research and safety profile that many people would like to see it. So I thought, well, let's take this stimulation practice back into an animal model where we can really look specifically at what's going on in the brain. Is it doing any damage? Is it doing any good? What sort of things can we find? But the problem is you have a machine that looks like this, and you have a mouse that looks like this, and it's a real problem to try and get some sort of equipment that can stimulate the brain in a focal manner, because I perhaps have omitted to say, but that figure of 8 coil delivers a really pointy focal stimulation in one particular area, which is great for depression when you know the brain area that you're targeting. But we can't do that in a mouse with this equipment. So this began a very long journey where we had to speak to the physics department at my university. We had to speak to the engineers. I got out my sewing machine and started winding little copper coils around on the sewing machine. And we went down this real rabbit hole with a very talented PhD student, Alex Tang, at the time. He was a PhD student and now is an independent researcher in his own right. And so we built these miniaturized coils that would be scaled to the size of the mouse head. And this is something that I never thought in my research career I would ever be doing. By a lot of work with different people, we bought ourselves a coil winder, eventually leaving my sewing machine behind. We worked with some computer models to develop models of electric fields. You wouldn't believe how complicated it is to measure a magnetic field, and even worse, to measure the electric field that then gets induced. There's a lot of computer modeling. And we ended up with essentially what would be the first dose-response study of these magnetic fields. Can you believe that over all the, I think, nearly 40 years now that TMS has been around, nobody has actually set out and done a dose response to see how much stimulation you need? So the coils we built, there are 3 of them. At the top, you have the low-intensity TMS, which delivers very low-intensity stimulation to the mouse brain. We have a medium-intensity TMS, which is probably about 10 to 20% of what a human would be getting. And the high-intensity TMS at the bottom is what you buy from the manufacturers. And they've built these incredible machines that they say will deliver focal stimulation to a mouse, but I don't know if you can even see the mouse under that giant big black box thing, which the commercial suppliers say, oh, it's very focal. And when you measure it, again, you're covering the whole head of the mouse, but at least it's not the whole body. We take our benefits where we can. So we essentially have this series of equipment that we can start to build up a whole series of basic research questions of what are these stimulation parameters doing to the brain? How is it working? How can we then use that information to drive plasticity in a way that can be beneficial? So this is now going through some of the research that I've done with others over the last 10 years. We can show that TMS changes behavior. We know that it changes how animals respond to certain tasks. And then we can show inside the brain that that's accompanied by reorganization of those particular pathways in the brain. We can go down an even lower level and say, well, we know that there are genes that are regulated. There are proteins that get upregulated, growth factors. You've probably heard of BDNF and things like that. But also there are processes like new neurons are born. We spoke briefly about stem cells, and it's really important to know that when you get new neurons born in the brain, they can integrate into pathways and start to promote plasticity and improve things like learning and memory. The excitability of neurons, that's a no-brainer really. When you're delivering electricity to the brain, you know that the excitability of the brain has changed. But one of the really fundamental things we found was that no matter how intense stimulation we deliver, even if it's very, very low intensity, we're changing calcium levels in the cell. Now that's really, really important because calcium signaling is central to a lot of different processes in all cells in the body. And so what that tells us is it's not just the neurons that are being stimulated, but all of the other supporting cells in the brain, like oligodendrocytes, like astrocytes, like microglia, all of those other cells are also potentially getting targeted by magnetic stimulation. And this is a real game changer, because traditionally we always thought that it was the neurons that did all the work, but we know in this room in particular that neurons are not the only ones, but that other non-neuronal cells play a key role. And so this is where the work with Kayleen came in. We got together many years ago and said, well, what's it doing to oligodendrocytes? And Kayleen took all of these ideas and put them together and came up with the absolutely groundbreaking finding that electromagnetic stimulation was somehow affecting oligodendrocytes. And she was the first one to discover that those oligodendrocytes can have their survival increase, they can be around for longer, potentially produce more myelin. And those results are really the foundation for the clinical trial that I'm sure you're all aware of. So a long story, a long background through engineers and physics and all sorts of things to get to the point where we never expected to end up. And it's been a really amazing journey to get to this point where some of the original research that I did has made a little contribution to some of the work that is now benefiting people here. So I'm going to talk about two things in a little bit more detail. I'm going to talk about neurogenesis, because everybody loves to hear about new neurons being born. When I was studying, I was told that once every pint that I would drink, I would kill neurons, and that was the end of it. But I can reassure you that neurogenesis does keep on going throughout life. And the glial cells also, and how those two things can hopefully come together. So the animal models that I initially started working on had to model some aspect of human depression. And people often say to me, well, depression is a terrible thing. It's very subjective. People self-report. The way that you diagnose depression is essentially asking somebody how they feel. And TMS is really a powerful treatment in this area because a lot of people do not respond to traditional medications, but those people tend to respond very well to TMS. So it rescues the population that doesn't respond to traditional medication. In order to try and make this better, what we want to try and do is find a way to treat some of the underlying causes of depression. And in animal studies as well as in patient studies, we know that the neurogenesis, the ability to produce new neurons, seems to be reduced in people with depression. And when you give antidepressants, it can increase the amount of neurogenesis, the new neurons that are born. So we had this amazing range of tools, of brain stimulation tools. We have access to a number of mouse models that can model some aspects of depression, so we thought we would go about trying to figure out what was going on in the brain and if we could affect neurogenesis with our treatments. So I know the burning question that you're all thinking is, how can we make a mouse depressed? Well, the short answer is we can't, really, because depression is a uniquely human thing. I'm not going to try and convince you that our mice are good models, but what I am going to try and convince you is that there's some things that humans experience in terms of anxiety, lethargy or hyperactivity, anhedonia, things that you just don't enjoy life anymore, and those are things that we can measure in a mouse. So although we can't get them to fill out questionnaires, we can look at the way they respond to stressful situations, we can look at the food they eat, we can look at their activity patterns, we can look at their response to really stressful environments, and we can get some indication of what's going on in their brain. So most of the models in animal models involve exposing these animals to stress for a period of time, and then measuring how their behavior changes after that exposure to stress. There are some surgical models where you can remove bits of the brain, but I'm not going to talk about those today. I'm going to mainly talk about a model where we've exposed mice to a chronic restraint stress. So this is our timeline, where essentially we expose mice to a period where they're put in a small chamber, so it's like a physical and almost like a social restraint. They're isolated in a box. And people compare these models to when you have a very stressful job, you get in your car every day, you drive for a very long time. It's a reproducible stress that happens every day, and you can't escape it. So the mice undergo this stress for 2.5 hours every day for 2 weeks, and we measure their behavior before and after that stress. And we can see that their activity patterns change. They have a higher stress response, and they no longer like or enjoy drinking sucrose water. So it's like they don't get any pleasure out of life after they've experienced that very long stressful period. We can then give them TMS or fluoxetine or Prozac as an antidepressant, so we have a positive control as well as our experimental treatment. And after this is a 4-week treatment period, we can then look at their behavior again and see whether the behaviors have improved. What we've also done in this study is specifically look at neurogenesis, measured the number of new neurons that were born. And we do that obviously by dissecting out the brains and looking in particular brain regions to identify where those cells are. So to cut a very long story short, our dose response was really interesting because if you look at in the vertical axis, you've got the low, medium, and high intensity. And then I'm going to show you 2 sets of results. First of all, for the behavior, the very low intensity wasn't very effective, but the medium and high were very effective. And as soon as you start to look at the neurogenesis, then you start to see a further difference where only the medium intensity was effective. So it's almost telling us directly here, more stimulation is not better. So we need to understand this dose response curve better, because although we may have good benefits on behavior by delivering medium and high intensity, the biological basis of those improvements is likely to be improved when we use medium, because it's actually changing the cells in the brain in a way that the high intensity is not. So how does this actually fit in with the research on MS that Kayleen and the Menzies Institute is doing? Well, myelination, we know, is also altered in people with depression. And a lot of brain scans will show you that the white matter is altered, and myelin overall in the brain is very much reduced. And there are particular areas where it gets particularly targeted. And so we thought, that's interesting. I wonder if that's happening in our mice. And so we looked at the mice that were receiving treatment with TMS, and we found that in the depression model alone, without treatment, they actually had reduced myelin as well. And, oh dear. Moving to the future. I probably pressed it too hard. Can we just go back a couple more, please? Oh, you're seeing my slides in reverse. 2 more, I think. Perfect. 1 more. Yep. So with that idea of myelination, we have in the mouse model of depression, we have reduced myelin. When we deliver TMS, it increases the myelin. And we have that matching with the results from Kayleen's lab showing that TMS can improve the survival of the cells that produce myelination. And this was a really unexpected finding where 2 completely separate research directions have come together. And it gives us a really interesting insight into how electromagnetic fields can affect the brain. They're clearly targeting myelination in some way. And also, it could be a new target for treating depression, that maybe by targeting myelination and improving that, it could also be a new way to approach depression therapeutics. So out of all of this have come a number of clinical trials, and I think before I've got a list of them on the next slide, but before I go there, it's interesting for me to see how the more I work in research, the more I realize that integration between the bench and the bedside is really, really crucial, because a lot of what I was doing on the bench was focused on the precise mechanisms of exactly what's going on in the cells. But as soon as you talk to people who are working in the clinic in neurology, they say, oh, have you thought of this? And then we can go back to the bench and actually ask that question. And the key component in all of this, obviously, is the person with the lived experience who can say, you know, this is really important to me. And I think this really means a lot to me. So can you look at these questions and can you explain to me why that is important? But fundamentally, that basic knowledge in the bench, for me, is a real driver of innovation, because you're not going to be able to get new ideas unless you create new knowledge. So it's not just this bench to bedside circle that goes round and round and round, but there's lots of other components that add to it. And today has been really inspiring for me, because I can see how much all of you contribute to each other's well-being, to the research that goes on, and to the hope for the future. The trials that I've been fortunate to be involved in include one for depression, where we're using that medium intensity stimulation, you know, convincing the clinicians to dial it down. You don't have to blast people's brains out, you know, nice medium intensity. And also the Taurus 1 and 2 trials, where again, we know that these particular intensities work. We need to reproduce those exactly in the clinic as we have validated in the lab. So altogether, this comes to producing a safety and evidence base, and also eventually to produce evidence of efficacy, which is where we're all heading, and we hope that we get there one day. And it's not just for depression, anxiety, and multiple sclerosis that I talked to you today, but other conditions like stroke, attention deficit disorders, even the sort of blue sky, that amazing city where we can have cognitive enhancement and improve the way that we learn and remember things. So this basic research gives us a platform to start to try and translate into all of those domains. So I'd like to thank you very much for your attention today and for your support for all of the research that goes on, that starts off in unexpected places, goes off into weird tangents, but eventually I think always comes back to benefit the translation of these findings into the clinic. And I really want to thank my research team that did a lot of the work that goes on behind the scenes and leaves me free to present all of their amazing work to you today. And particular thanks to the funding bodies here, because without their support, obviously, we couldn't do the research that we do. So thank you very much.
Another outstanding presentation, and I can honestly say, Jenny, I learned more out of that than 3 days in New York. So incredibly well done. And I know that Rowan has written your name down, because, of course, MS Australia's conference is at the end of the year, and I think that would fit beautifully in MS Australia's conference. Any questions while we've got Jenny up there? Remembering, of course, that this is world-leading research. So, one question. Everybody's wondering what happens to the mice afterwards. That's what everybody's thinking.
Well, my question's not about mice. But right at the beginning of your presentation, you mentioned physical activity and how that's really one of the most effective ways of improving plasticity. Are you aware of any other studies that look at remyelination from that physical activity point of view?
I'm not, actually. Maybe, I don't know if Kayleen's aware of that question. I'm hoping she might be able to answer that.
Yeah, so there is some evidence that I guess increased running or activity can actually increase not only neuron addition to the brain, but also support new oligodendrocytes, so the addition of new myelin. But also at this point, some of those studies have been a bit variable, and so I don't think we're at the point where we could say this is the right amount of activity to promote myelin addition. It's like Jenny's saying, everything sometimes is about how much is good and how much is not so good. But generally speaking, for brain health, physical activity is a positive.
And in any form?
Yeah, so different people can have different levels of a physical activity. Running is one of the things that is studied the most, but different levels of physical activity will activate these sorts of processes to differing degrees.
Bruce?
Thanks, Jenny. I just had a comment that when we're talking about remyelination in humans, it's a lot more difficult to measure it than in mice. We have a problem with sacrificing humans, and so it's a minor ethical issue. So we are developing the mechanisms to measure remyelination in humans, and it's a lot harder, and that's why we rely so heavily on the animal models. And thanks for a fantastic talk, Jenny.
Yeah, thanks, Bruce. And the MRI techniques are getting better and better. I think the resolution and the analysis quality, so it's really exciting now to be able to map MRI onto anatomy, because you can also put a mouse or a rat in an MRI machine so that you can understand what's going on, in the tissue, in the MRI scan, and use that to help inform the humans. So I think that interchange between the research streams is really important.
Perfect. Round of applause, please, for Jenny.